All right. Picking up where we left off, I was talking about you can calculate the variance in a particular trait, and you can try to divide that variance into portions of the phenotypic variance that are due to genes and portions of the phenotypic variance that are due to environmental effects. And you can define something called broad sense heritability of a trait as variance due to genes divided by total phenotypic variance. Here's the thing, you can have traits that are genetically inherited, but in a given population, they might not have any genetic variance at all. Actually, a really good example would be those inbred mice that I was talking about earlier, those strains of mice that are propagated by the Jackson laboratories and used in biomedical research all over the world. Those strains of mice have been so inbred for so long that for all practical purposes, they're clones of each other. Um, in the species Mus musculus, the domesticated mouse, as a whole, we know the genetic basis for a lot of traits. So fur color, for example, is genetically largely determined. And we know a number of genes that govern fur color in mice. In an inbred strain of mice, though, the broad sense heritability of fur color is zero, simply because there's no genetic variation at all, because all of those mice are, for all practical purposes, they're all clones. They don't have any genetic variance. And if they're all genetically identical and they're all raised in the same environment, they would have no heritability at all, because there's no genetic variability, no environmental variability, there's no phenotypic variability at all, because those mice are all the same. So you can have a trait that is under genetic control, but within a population, it has no heritability, even though the trait is inherited in the sense that it's genes that determine what that trait is. This is important when you're referring to heritability of a trait. It's always in reference to a particular population. We can break down genetic variance a little bit more into additive genetic variance, dominance genetic variance, and interaction genetic variance. When we're talking about additive genetic variance, that's the amount of genetic variance that depends only on the number of alleles. Our white spotting was a pretty good example. White spotting in cats, you could measure that trait, and you could measure, let's say, if we wanted to make it quantitative, you could measure the percentage of the cat's body that had white spots. And let's see, your average might be, I don't know, the average trait for a cat with no white spotting genes would be 0% of the cat has white spots, and a heterozygous spotted cat might average 33% white spots, and a cat that's homozygous for the spotted gene might average 66% white spots, or something like that. In that case, the amount of white on the cat's body does vary, so there's phenotypic variance. The variation is primarily due to genes, and more importantly, it's additive genetic variance, because what it depends on is simply the number of alleles. If your cat has no copies of the spotted gene, it has no spots. If it has one copy of the spotted gene, it has, let's say, 33% spots. If it has two copies of the spotted gene, it has 66% spots. There's a perfect linear correlation between the number of copies of the gene and the number of the amount of body that's covered in white spots. That's, in that case, the genetic variance is entirely additive genetic variance. Some of our genetic variance, however, in another case might be due to dominance effects. And you can also have variance that's due to interactions between genes, because genes can modify each other's expression. Genes can, a gene at one locus can cover up the effects of a gene at a completely different locus. An example from cats would be that we have this white spotted gene, but there's a second gene on a completely different chromosome that if the cat is homozygous for it, the cat will simply be completely white. And in that case, you cannot see whether it's carrying any white spotted alleles or not, because the presence of white spots is completely covered up by this completely different gene that makes the entire coat white. That is going to be a source of experimental error, by the way, in the lab that you've done or will do, because some white cats are white because they're homozygous for the spotted alleles, and the spots are so extensive that they cover virtually the entire cat. But there's other cats that are white because they're homozygous for a completely different gene that simply turns them all white, whether they're carrying white spotted genes or not. So you can't really tell by looking whether an all white cat has the spotted alleles or doesn't, and that is going to introduce some error into our results. It's just one of those things. But that's a case where phenotypic variance is caused by interaction between genes. In this case, it's one gene covering up the effects of another. That happens. I want to work out an example for you, and I'm going to go to the whiteboard for a second. This is the scarlet tiger moth. I believe that's native to Britain. And it's got spots on its front wing, and the number of spots is governed by a single gene that has two incompletely dominant alleles. So up at the top, a moth that is homozygous dominant has lots of light spots on the front wings. In the middle, that's a heterozygote that has fewer, and below, that's a homozygous recessive that has even fewer. So the homozygous recessive has two big white spots on each forewing, 1, 2. The heterozygote has, I'm going to say there's 4 prominent white spots on the front wing. And the homozygous dominant, I'm going to wave my arms and say that that's equivalent to 6 white spots on the wing. Okay, we can quibble about how big a spot has to be, or whether 2 spots that are sticking together count as 2 or as 1. By the power vested in me by the state of Arkansas, I'm going to say that that top one has 6 spots on the wing. And the reason I say that is I want to go to whiteboard. Okay, right. So we have these moths. I trust everybody can see what I'm typing. So we have these moths. And if you're SS, you have 6 spots. And if you're a heterozygote, you have 4 spots. And if you're little s, you have 2 spots. So let's say we've got a whole bunch of moths flying around. We've got a population, and they vary in spot number. And we can, catch a bunch of moths and count them, so we could measure the variance in the population. All of this variance is due to additive genetic variance. What makes me say that? I'm going to draw a graph. That's pretty thick. Okay, on the x-axis, I'm going to put mean parental phenotype. And on the y-axis, I'm going to put mean offspring phenotype. Okay, let's think about this. Suppose we take two moths that are both little s. Both of them have two spots, right? Okay, I think that's what I want. Two parental moths, and they're both little s. Both of them have two spots. So the mean number of spots is going to be 2, right? It's the average of 2 and another 2. All right. The offspring are all going to have 2 spots. So however many babies that these moths have, all the babies will also have 2 spots. So we can put a point right there. Okay, where the mean parental phenotype is 2 and the mean offspring phenotype is also 2. Let's say we cross two big S moths. The mean parental phenotype is going to be 6. All of the babies are going to have 6 spots if you're crossing two homozygotes. So the mean offspring phenotype is also going to be 6. So there's another point. Let's say we're crossing a big S and a little s parent. The babies are all going to be heterozygotes. So if one parent has 6 spots and the other parent has 2 spots, the mean parental phenotype is 4, the average of 6 and 2. All the babies are going to be heterozygotes and they're going to have 4. So the mean offspring phenotype is going to be 4. That's another spot. And I'm not going to do every possibility, but we could keep going. Let's say we cross a big S moth and a big S moth. Well, one parent has 6 and the other parent has 4. So the mean parental phenotype is 5. And if you cross a big S and a big S moth, right, half the offspring will be homozygous dominant and have 6, and half the offspring will be heterozygotes and have 4. So the mean offspring phenotype is also going to be 5. If we cross big S and little s, the mean parental phenotype's 3, the mean offspring phenotype is 3, and so on. What we'd get is a line looking like that, right? And the slope of the line, well, the equation of the line is basically y equals x. The mean parental phenotype is always going to be the same as the mean offspring phenotype. And the slope of that line is 1. The way we estimate the additive genetic variance for a trait in a population is we take the mean parental phenotype, we graph it against the mean offspring phenotype, and we look for the slope of that line. The slope of the line is the way we calculate narrow sense heritability, the amount of variance in a trait that's due to additive genetic variance. Now, if we had a different system where we didn't have this kind of incomplete dominance, where we had dominance effects, we actually would not get a line with a slope of 1. We'd have more scatter, for one thing. And for another thing, we'd end up with a line with a slope of about 0.7 or something like that, if we were working with a different species Dominance would mean that we didn't have this kind of slope. So in this case that I've just worked, the additive genetic variance is, the additive genetic variance is 1, meaning 100%. No, not the additive genetic variance. The narrow sense heritability in this population is 1. And what that means is all of the variance in the population, all of what is making our moths look different from each other, all of the variation that we see in the number of spots on the wing is due to genes that have additive effects. 100%, the number 1. Okay, hopefully that was not impossibly unclear, because I want to cut over and show you folks what I got when I looked at your data. Okay, so there it is, spring 2021. These are the numbers that you sent me, and I'm assuming, I'm sure I'm right, you have no incentive to lie. I'm assuming all of this is accurately measured. Down here, by the way, I calculated the variance of my total data set. You can see I've got data going back to spring 2012. We'll go back and look at those graphs in a minute. But we've got data going back almost 10 years, and the variance in parental height is 5.4. It's greater for the offspring data set. It's actually 12. There's more variance in the height, the mean height of parents, than there is in the mean height. Come on, Wagner. There's less variation in mean parental height than mean offspring height. I'm guessing that's because every mean parental height is an average of only 2 numbers, whereas you can get a lot more variance if you're not, if some people are only children and some people come from big families with many siblings. That might account for why the mean offspring height has greater variance. Anyway, if you look at the graph over here, this is what I got when I graphed your data. There's a lot of scatter, but we measured the slope of the best fit regression line. And let me see if I can make that look bigger. Label options, text options, text outline. Okay, there's a bunch of fancy stuff here, but I don't see a way of increasing the font size. Can I do this? Can't do that either. The slope of the line going through them is almost exactly 0.7. It's 0.7027. It's this dashed line right here on the chart. So what that's telling me is 70% of the variation in your heights, you, the evolution class in the spring of 2021, 70% of that variation is due not just to genes, but to genes that have additive effects, like the gene governing the number of spots on the wing of a tiger moth. That's actually pretty close to the published values. This is what I would expect. This has been estimated for a lot of human populations, and the heritability of human height is usually calculated at about 0.7. It's usually not worthwhile calculating it to more than 1 decimal place, but this is pretty much, we basically got the expected answer there. And if I look at all of our data, this graph is what it looks like for everybody since the spring of 2012. That's our complete data set. You can see all those points. You can see the regression line right there. And I don't know if you can see that equation, but it's y equals 0.8338x plus 11.306. So the slope of that line is 0.83. So based on a much larger data set, based on over 150 students that I've had over the past 9 years. That's our estimate for the narrow sense heritability of human height. In the student population at UCA, 83% of the variance in height can be attributed to the effect of genes that have additive effects. And again, that is close to the usual answer that you get is 0.7. It's a little bit high here. Maybe in 10 years, it'll have gone back down once I've doubled the number of data points. But it's definitely in the ballpark. If there were no correlation between mid-parent height and mid-offspring height, we just have a cloud of points. And if you have, basically scatter data like that, then the slope of the regression line is 0. But as it is, we've got a pretty sizable slope of the regression line, and that's our estimate of height's narrow sense heritability. By the way, I did a couple of bar graphs. Mid-parental height, you can see, has a bell curve. A few people have parents that average very short. A few people have parents that average very tall, and most people's parents are somewhere in the middle. This is true for mid-offspring height as well. Some people come from families where they and their siblings are really short. A few people come from families where they and their siblings are very tall. That highest over there was 80 inches. That's what, 6'8"? I'm trying to remember who that guy was. He probably played basketball. But most people come from families where the mid-offspring height is somewhere in the middle. So both of these are pretty close to that bell curve or normal distribution. All right. Checking the, just checking the chat. Doesn't look like there's anything I need to take care of immediately. Okay, right. I'm going to go back to the slides. The reason this actually has practical importance is you can plug it into this. R equals lowercase h squared times s is called the breeder's equation. And it is practically important because you can use it to predict whether selection will be effective in changing the mean value of a trait in a population. Capital R is the response to selection, and that's the difference between the mean trait in parents and mean trait in offspring. Capital S is the selection differential. That's the difference between the mean trait in the general population and the mean trait in a population that's selected for breeding. Okay, what the heck do I mean by that? I'm going to go ahead and go back to whiteboard. So there we are. Let's clear that. All right, let's say I'm a chicken farmer. Okay, this is Arkansas, so it's pretty believable. And I've got some chickens, and they are laying eggs, let's say. And some of my chickens are extremely lazy and only pop out the occasional egg when they feel like it. And some of them are absolute egg laying go-getters that are just, want nothing more in the world than to crank out as many eggs as humanly possible. And most of the chickens are somewhere in between. Okay, so I could take egg production. We could measure that in, I don't know, number of eggs per week or something like that. And I would find that a few of my chickens weren't laying or weren't laying very much. I don't want the line, I want this. There. A few of my chickens were very lazy. They're not laying many eggs. Most of my chickens are kind of in the middle. And a few of my chickens are seriously busting the curve because they're cranking out eggs on some kind of internal factory. So on the y-axis, we have number of chickens. Okay, so most of my chickens are kind of in the middle. A few of my chickens are complete slackers. A few of my chickens are absolutely insane egg producers. And naturally, what I want to do is get more eggs because that way I make more money. So I want to selectively breed my chickens for maximum egg production. Okay, how do I do that? Well, right here, we've got the mean, and because it's the mean, we call this X bar, X with a bar over it. If this distribution is truly normal, then the mean, the median, and the mode are the same thing. And that means that right in the center of that bell curve is where the mean egg production is going to be. All right, let's say that I decide that all of my chickens that are producing more eggs than this cutoff, they get to live. All of the chickens that are in here and are not producing enough eggs end up in Kentucky Fried Chicken baskets. All right, end up as part of a 12-piece drumstick basket or bucket, I guess, at KFC. So all of the chickens that are producing fewer eggs than this end up eaten. All of the chickens that are producing more eggs than that cutoff line, they survive and they get to, they get to have babies, and they get to be the founders of the next generation of my flock. All right. Well, for this batch, I could calculate for these chickens over here, or the survivors, I can also calculate the mean number of eggs that they're laying. And I call that X bar with a star. So there, X bar star. X bar is the average number of eggs that my chickens are laying. X bar star is the average number of eggs that my surviving chickens are laying after I've culled all of the lazy chickens and sent them off to be made into original and extra crispy. With me there, I trust. Okay. I can define a number called S, that's the strength of selection, and that is equal to the absolute value of the absolute value of X bar minus X bar star. The greater that difference is, the bigger the gap between X bar and X bar star, the larger is my selection differential. If I'm allowing most of my chickens to survive and I'm only trimming off the slackers way back here, then there's not going to be a big difference between X bar and X bar star, between these two means. On the other hand, if I'm getting rid of 99% of my chickens and I'm only allowing a very small number of very high producers to have any offspring in the next generation, then that gap between X bar and X bar star is going to be greater. There'll be a greater difference between the mean phenotype before I started selection and the mean phenotype after I've done the selection and culled all of my poor layers and sent them off to, end up as McNuggets or whatever it is one does with excess chickens. So I can define this thing called capital S as the strength of selection. Okay, now what? Let's say that, let's go back to lines. I take the chickens that are above the survive line, only the ones that lay the most, and I'm going to draw another set of axes here. All right, I take the surviving chickens, I let them breed, I let them have baby chicks. Actually, I don't want a different set of axes. I'm going to go ahead and do this on the same axis, but I'll make it a different color. Okay, after I have done my selection, I take these survivors, I let them reproduce. I take my survivors, I let them have babies, and in the next generation, I presumably will get something like this. After one generation, I should see something like that. I'll still have a population where there are some individuals that aren't cranking out huge numbers of eggs and some that are complete overachievers, and most are going to be in the middle. So my average is going to be right there. And I can take that average after I've done selection, and I'm going to go ahead and call that, I need to erase some of this. Oh, I didn't mean to erase all that. My average before selection was X bar. My average after selection I can call Y. Where's my line tool? There. I can call Y bar. Okay, so Y bar is the average egg production per chickens after one generation of selection. After the surviving chickens have all been allowed to grow up and have babies of their own. It's the effect after one generation. So we defined the selection differential is equal to the absolute value of the differences between X bar and X bar star. Draw the bars. Okay, the response to selection is equal to the absolute value of the difference between X bar star and Y bar. Let me draw those bars in there. Okay, so my selection differential is the difference between the mean before selection and the mean after I've culled. The response to selection is the difference between the mean phenotype of the culled population and the mean phenotype after one generation of reproduction. It's the distance over which I can shift that curve. If selection has no effect, then no matter what I do, that bell curve after one generation, that purple bell curve, is going to lie right on top of the red bell curve. On the other hand, if my flock responds to selection very strongly, then that purple bell curve is going to shift quite a bit compared to the red bell curve with which we began. So I have my strength of selection, I have my response, and the breeder's equation is R, the response to selection is equal to narrow sense heritability times the strength of selection. So what this means is that if we have some estimate of the narrow sense heritability of a trait, what we can do with that is predict the response to selection. We can predict whether or not selection on a population is going to have a measurable effect on it. We can get some idea of whether it might be worth doing or not. To give an example, I read somewhere about a turkey farmer back in America in the 19th century who got the idea of putting little turbans on the heads of all of his turkeys. The idea was that he hoped to eventually breed a variety of turkey that was born with little turbans on their heads. Because haven't you always wanted a turkey with a turban on its head? You know, I'm sure, you know, that's going to be the hot new Christmas toy, I am certain. Obviously, this did not work, but the reason that it didn't work is that wearing a turban on your head has a narrow sense heritability of 0. You know, some people wear turbans on their heads and some people don't. Whether or not you wear a turban on your head has nothing to do with additive genetic variance. There are no genes that make it more likely that you're going to wear a turban on your head. Okay, that is entirely down to personal choice and the culture that you're raised in. You know, some people grow up in cultures where it's expected that people wear turbans, or at least men. Some people grow up in cultures where nobody bothers with a turban, but there aren't any genes that encourage you to wrap long strips of cloth around your head. That has nothing to do with your genetics. That's a matter of your choice and a matter of the culture that you come from. And I assume it's the same in turkeys. So the narrow sense heritability of wearing a turban on your head is absolutely 0. So that means the response to selection is going to be a 0, no matter what the strength of selection is. You could put turbans on the heads of all of your flock of turkeys and then kill all of the turkeys except for the few that wear it the best, maybe look nicest in a turban. You know, you can select that flock for turban wearing until you're blue in the face, and it will have absolutely no effect because the narrow sense heritability of turban wearing is 0. So the response to selection is always going to be 0, no matter how strong your selective regime is, no matter how stringently you select your breeding turkeys. I've used kind of a goofy example, but in slightly less goofy examples, this is used by livestock breeders today, this breeder's equation. So mercifully, we'll go back to the slides. And there's the breeder's equation again. Narrow sense heritability, that h lowercase h squared, is good for predicting whether or not selection will be effective in changing the mean trait value of a population. So that's some old data for spring of 2012 and summer 2013. For that data set, we got a slope of 0.9 as our estimate of narrow sense heritability. The accepted value was 0.7. That's what our class basically got. What this means is that if I wanted to take UCA students and I don't know, kidnap everybody, put them on an island, and set up a fiendish breeding scheme to breed a master race of NBA centers or something like that, at least at the beginning, it should succeed. Because if I took a whole bunch of UCA students, did something horrible to most of them, and only picked out the ones that were tallest to be the progenitors of my new master race of basketball forwards, at least at the beginning, it should work really well, because that narrow sense heritability is very high. And that means a strict selection regime where I only pick the very tallest should give me pretty strong results. I should get a next generation of students that actually are quite a bit taller than the population that they came from. I'm not actually going to do such a thing, be it said. Does make for sound like a very bad science fiction movie plot. This is some data from a wild population. This again comes from the work of Peter and Rosemary Grant, who have been studying Galapagos finch populations for going on 50 years now, and tracking changes in those populations from year to year. They were able to measure mean parent versus mean offspring beak size in these Galapagos finches, and the slope was about 0.8. So 80% of the variation in beak size in Galapagos finches is due to the additive effects of genes. You'll notice that in 1976, you got one line, and in 1978, you got a different line. The line was shifted. Well, it looks like it's shifted down. I think it's actually shifted to the right, but that slope doesn't change. The dark spots show you data from before a drought, and the open spots show data from after a drought. What that's telling you is that average beak size did shift in that population from before to after the drought. And that makes sense because in the drought, the finches that had small beaks tended to die, and finches with large beaks tended to survive. So that line shifts, but its slope doesn't change. And that suggests that we are, in fact, measuring something that is strongly influenced by genes. A problem that results. If you thought this was bad, it only gets worse. One of the complications of studying variance is that there is some variance that results from interactions between genes and environment. So really, total phenotypic variance is the sum of genetic variance, environmental variance, and environment gene interaction variance, written VG cross E. And a terrific example of this is something that we call norms of reaction. When you start studying genetics, we start assuming that genes are really the only thing that matters. That, you know, if you cross two purple flowered pea plants, you'll get only purple flowers in the next generation. Or if you cross a purple and a white, you get only purple in the F1 generation, but purple and white together in the F2, or whatever it might be. We tend to assume that this is going to be true, no matter, no matter what the surrounding environment is. We tend to assume that genotype determines phenotype. That if a pea flower has two copies of the purple allele, it will look purple. And if it has two copies of the white allele, it will look white. End of story. What makes things more complex is that a genotype very often does not specify exactly what the phenotype is going to be. Instead of one genotype determining the phenotype, a genotype often determines a range of phenotypes that an organism might take. And then you can think of the environment as determining exactly which phenotype in that range the organism ends up having. So you can think of the genes as determining the range and the environment determining what actual phenotype you've got. When phenotypes vary with environment, we call this phenotypic plasticity. And we can calculate the specific relationship. We can calculate functions that show the relationship between phenotype and environment given a certain genotype. And we call that a reaction norm, or a norm of reaction. That's a lot of words. Let me just show you. This is two pictures of a microscopic animal. It is, in fact, multicellular, made of several hundred cells. Belongs to a group called the rotifers. Very, very common in fresh water. There's also lots of them in the oceans. If you've ever looked at pond water under a microscope, you'll probably find rotifers. I know I spot them every time I get pond water from Lake Conway or Lake Beaver Fork. They're all over the place. They are quite small. You can see that scale bar is 50 microns. So the total length of one of these is maybe 150 microns. I don't know if any of you folks are, I guess all the people who took invertebrate zoology from me last time I taught it have probably graduated. But I think I'm going to be teaching that in the spring of 2022. So if you're still around, you might want to take that. We'll talk all about rotifers there. Okay, anyway, rotifers of this species, Brachionis caliciflorus, when you grow it in an environment without predators, it has this kind of vase shape, and that's what you see on the left. Looks like a little swimming vase with little toes down at the lower left. If you grow it in the same environment as a larger rotifer called a splanchna that eats it, it sprouts spines. And on the right, you can see a Brachionis that was grown in close proximity to a splanchna. Brachionis will even do this if you expose it to water that a splanchna has swum in, but not actual predators themselves. If you just give them water that smells like predators, they respond to some sort of chemical cue. I'm not entirely sure what. And they'll sprout spines, like you see in the individual on the right. That's phenotypic plasticity, right? The phenotype is not strictly determined by the genes. The phenotype can change in response to some kind of environmental signal that the rotifers get from the presence or absence of predators in the water they're swimming in. There's other critters that do this as well. This is Daphnia. We do lab experiments with Daphnia in 1400. I'm not sure if you folks do. Daphnia is a little crustacean. The common name for it is the water flea. It is pretty common in many bodies of fresh water. It's sometimes collected as live food for fish. So if you've ever kept a freshwater aquarium, you might have come across Daphnia. And this is cool. On the right is an ordinary Daphnia. This picture was taken through a scanning electron microscope. And on the right is a normal Daphnia. On the left is Daphnia that's grown in the presence of predators. I think in this case, the predators would be some insect larvae. And this species of Daphnia grows a helmet. The head's got a big spike on it. The tail spine is a lot longer. This makes the Daphnia longer and spikier than the predator likes to put up with. And again, you don't have to have actual predators in the jar with your Daphnia. The predators produce some kind of chemicals, and the prey responds to the chemicals and sprouts these horns. The chemicals are called pheromones. So this is phenotypic plasticity. To be specific, it's called an inducible defense. We actually see a lot of examples of this. Clover. When you start damaging a clover leaf, the clover leaf will start producing cyanide. And not just that clover leaf, but surrounding clover leaves will begin cranking out cyanide as well. So clover doesn't produce cyanide 24/7, probably because it's not metabolically efficient to do so, but it can turn on that defense when a clover plant or a neighboring clover plant is attacked by a predator, like a caterpillar or whatever it is that eats clover. So that's an inducible defense. One nifty example is a European wildflower that grows on the edges of streams, Ranunculus aquatilis, or water crowfoot. It has stems and leaves that are submerged and stems and leaves that grow above the surface of the water. And you can see the leaves that grow above the surface of the water are very broad, but the leaves that are growing below the surface of the water are filament-like. They look like little bundles of branching filaments. So in the same species, even on the same plant, the phenotype of, what do the leaves look like is phenotypically plastic, right? These two, the broad leaves and the filament leaves, have the same genes because they're on the same plant, but which phenotype you get depends on the environment. So the genotype must specify a range of possible ways you could build a leaf, and then the environment determines which of those ways a given plant is going to adopt. So that's your phenotypic plasticity. I neglected to include it in the slides, but next time if any of you folks garden, or heck, even if you don't garden, but I've seen displays like this in the floral section of Kroger, for example. If you go grocery shopping, there is a garden plant around here that a lot of people like. It's called the hydrangea. And hydrangeas produce these big bundles of flowers that can be either pink or blue. And it turns out that pink versus blue hydrangeas is yet another example of phenotypic plasticity. It ultimately depends on soil pH. And now I can't remember which way it works, but I think if you grow your plants in neutral soil, you get blue. If you grow your plants in acid soil, you get pink. The ability of that plant to make blue pigments depends on the soil chemistry. If the soil chemistry is one way, then the plant is not able to make blue pigments and it ends up looking pink. You can turn a pink hydrangea to blue by putting iron nails among the roots. That changes the soil chemistry enough that the plant will start producing blue pigment and will actually turn blue. And I had a terrific picture of this that I have forgotten to insert in the slides. I might do it when I might add it and then print up the handouts. But that's an example you can see, and you probably will be able to see this spring because this is about the time of year when Kroger starts putting hydrangeas in its floral section. I know this because I usually get some for my wife because she likes them very much. And you'll be able to see pink and blue hydrangeas. That's not due to a genetic difference. The plants are genetically practically the same. It's due to the environment affecting gene expression. All of those plants have the genes needed to make blue pigment, but the environment determines whether those genes can operate or not. That's phenotypic plasticity for you. Here's what I meant by a norm of reaction. There are, okay, the y-axis, for some reason, this illustration is captioned in German. But on the y-axis, it says Anzahl der Facetten. That means number of facets. Somebody, probably a grad student, calculated all of the little facets in the eyes of fruit flies. Sat there and counted them. Yeah, definitely a grad student there. And if you look up at the top of this graph, there's a black line and it's labeled wild type, or okay, Wildtyp, but it's wild type. And you can see that if the flies are grown at 15 Celsius, that's what, about 60 Fahrenheit, you end up with over 1,000 little facets per eye. But as you grow these flies at progressively higher temperatures, they develop a lower number of facets. So if you grow the same wild type flies at 30 Celsius, which is what, about click, roughly 90 Fahrenheit, maybe 88, 90, those flies only have about 700, 750 facets. Okay, that's phenotypic plasticity. The number of facets in the eye of a fruit fly is not genetically pre-programmed. It depends in part on the environment that the fly grows up in. So if you raise your flies in cold weather, they'll have more facets. And if you raise your flies in hot weather, you have fewer. That is phenotypic plasticity. That specific relationship, right? The line that we've drawn on this graph is the norm of reaction. Okay, the norm of reaction is a mathematical description of phenotypic plasticity. So if you look at the others right here, there's two different fruit fly mutant alleles called infrabar and ultrabar. Bar eyes in fruit flies are unusually small eyes that are shaped like little bars instead of shaped like, the big globes that flies typically have. And bar mutants in fruit flies have been known for some time. Flies that have the infrabar, I guess they're homozygous for the infrabar gene. If you raise them at low temperatures, they might have about 200 facets per eye. If you raise them at higher temperatures, they might get up to maybe 300. On the other hand, if you've got flies that are homozygous for ultrabar, if you raise them at cool temperatures, you'll get about 200 facets. But if you raise them at high temperatures, you get fewer than 100. You get maybe 70 facets per eye. So the difference between the infrabar allele and the ultrabar allele isn't that they necessarily create different phenotypes. You can see where those lines cross. If you raise fruit flies at a temperature of about 17 Celsius, infrabar and ultrabar flies will look the same. They'll have identical phenotypes if you raise them at the point where those lines cross. What's different is not that infrabar and ultrabar genes always produce different phenotypes. What's different is the fact that they've got different norms of reaction. Flies with the infrabar genotype and flies with the ultrabar genotype respond to different environments in different ways. We can represent that mathematical relationship between temperature and facet number by those lines, right? We could work out an equation for graphing those lines. And the equation of those lines would be the norm of reaction. The norm of reaction is the mathematical relationship between phenotype and environment. Okay, everybody happy there? Classic example, all the way back in 1948. Couple of botanists in California went out and collected wild plants in the genus Achillea, yarrow. Don't know if you know yarrow, but you can grow it as a garden herb and sometimes add it to salads. It's got these fringy leaves that have pleasant taste if you like salad. There's cultivated yarrow that you can, I don't see it a lot around here, but you can grow it. It's a perfectly nice plant. So they went out and collected 7 wild yarrow plants, and yarrow happens to grow very nicely from cuttings. And they grew 7 wild yarrows in the same garden at a site in California called Mather. And here's what they got. You can see that plant A did very well. Plant A grew to a height of 70 centimeters. That's a centimeter scale at the left. Plant G only got 20 centimeters. It did not do very well. E did pretty well. B didn't do so well, and so on. They're all growing in exactly the same environment. So variance in this little population due to environment is 0. So all of what is making these plants look different must be that they've got different genes, right? Because they're in what we call a common garden experiment. They took cuttings from the same 7 wild plants and grew them at a different garden at Stanford in the bustling metropolis of Palo Alto, back before Silicon Valley killed it. And they got this. There again, all of these plants grew in the same environment, so the differences in height must be genetic, right? And again, plant A grew very tall, grew 120 centimeters. That's what, roughly 3 feet and change. That's a pretty tall plant. Plant G did not do so well. B did very well, over 100 centimeters, and so on. They're all growing in the same environment, but the height differences must be genetic, right? Because the environment's the same. But if you compare the two directly, you get this. Mather is up at a high elevation, by the way, and Stanford is down pretty close to the ocean. So you've got different climates. The Mather and the Stanford plants are genetically identical because they both grew from cuttings from the same 7 original wild plants. So plant A at Mather and plant A at Stanford are genetically the same. Plant B at Mather and plant B at Stanford is genetically the same. C at Mather and C at Stanford are genetically the same, and so on. And then, of course, all the plants at Mather have the same environment. All the plants at Stanford are growing in the same environment. And here's what I want you to see. What's different about these 7 different plant lineages is their norms of reaction. Okay, A is always the tallest in either environment. But look at B. B does not grow very tall at high elevation at Mather, but it comes in second. It's almost as tall as A at Stanford. Okay, there's E right there. E does not come in very tall at Stanford. It comes in second up at Mather, right? G doesn't amount to any. G is the lowest in both, but you can see G is really stunted at Mather. It's not nearly as badly affected at Stanford. D does not do very well at Mather, but it does much better at Stanford, and so on. What's different about these 7 different plants is not so much their genotypes, it's that they've got different norms of reaction. It's that they respond to different environments differently. Ta-da! And that means that, a plant that does really well in one environment is not necessarily going to do really well in a different environment. Last thing I want to do is give you a case of how this might apply to human behavior. This was a study from 2002. I haven't seen follow-ups, although I'm sure there must be some. But I checked a few years ago, and this still seems to be a valid result. Am I going over? Shoot, it's 12:05. I'm going to go ahead and stop there. That's actually a pretty good place to stop. So we'll stop with our consideration of yarrows. And this coming Tuesday, I'll try to tie this in together and get back to the bell curve. And then I think I can explain why the bell curve has very much gotten on the wrong foot. So I will hereby stop the recording.